Okay, can I get someone to confirm you can hear me? Once again, I've got a error message saying my speaker isn't working.
We can hear you.
Thank you. Before we begin, are there any questions? All right, let me state that I have graded, I think it was Lab 5. Let me check on that exactly, but I did get that graded on Wednesday, and if you did not get 100%, which most of you did not, you can get it corrected not by this Saturday, but a week from this Saturday. Let me go to make sure that that's Lab 5. I'm not sharing the screen, am I?
No.
Thank you. I'm looking at the grade book. That's why I asked. I didn't want to share the grade book. Yes, Lab 5 has been graded. If you didn't get 100%, you may submit corrections. And normally I would make it this Saturday, but I didn't get it graded until Wednesday, so I decided to give you extra time. Let me shut down the grade book and then I'll share my screen. Where am I here? Don't want that. Oh gosh, I just logged out of my I got to do this whole thing over again. Sorry about that. I just logged out of, oh, come on. I just logged out of Canvas by mistake and here we are. Fortunately, I didn't have to get Microsoft to verify my existence. Um, all right, normally it's gotten so bad that every time I try to log into either my email or to Canvas that I have to have Microsoft verify my existence. And when you're getting into Canvas for the first time, you got to get Microsoft to verify your existence twice because you have to get an email to log into Canvas. All right, if there's no further questions, let's begin. Sharing my screen now. So today is August 8th. Boy, that's the wrong syllabus I've got up here. Today is August 8th, and we should be working on Chapter 6. Hopefully, we'll finish Chapter 6 today and get started with Chapter 8. There is a lab today. I'll talk about Lab 8 in the lab time. Any questions about what we're doing? Well, that didn't move. Not seeing two here. There it is. All right, I have that we were on this slide the last time. Oops, didn't mean to move that. Trying to move this. this slide the last time where I talked about streaking for colony isolation, which a number of you got wrong in the lab. The major goal for streaking for colony isolation is simply to get isolated colonies. It is not to get a pure culture. In fact, the first time you streak for colony isolation, about 30% of the colonies will not be pure colonies. They will be colonies from a mixture of cells. When we're talking about microbial growth or bacterial growth, remember this refers to an increase in the number of cells, not an increase in the cell size. The mechanism of growth by bacteria is most bacteria divide by binary fission, the most usual method for bacteria to reproduce. We'll talk more about that in a little bit. But there are a few bacteria that multiply by budding. A cell will form a small outgrowth called the bud. That bud will enlarge, and then it will separate from its parent cell when the bud becomes almost the size of the parent cell. And then there are a few bacteria species that reproduce by reproductive spores. These are spore tips at the filaments, meaning usually they're bacteria cells that are in filaments, and then they're more likely to scatter from either an animal moving by or from the wind. So let me draw that out. So we usually have a parent cell, and it will put itself standing up. So this would be like the surface. And then it'll make reproductive spores on the tip of the cell. Usually there's about 4, but there could be more, like 6, depending on the species. Nope, I drawn 5. I meant to draw 4. Oh well. And what happens is one of these spores will go elsewhere. Let's go here, and then the reproductive spore will germinate and become a parent cell, which will then enlarge. Maybe not as large as the parent cell, but come on, mouse. And then it'll go through normal metabolism and perhaps reproduction, which could occur by reproductive spores. Any question about any of that? So those are the 3 ways that bacteria cells can reproduce. Depending on the species, most species will divide by binary fission, but a few by budding or by reproductive spores. Remember, reproductive spores is where one parent cell makes several reproductive spores. So if they make 4 reproductive spore, one parent cell then becomes 5 cells. Is the parent cell still around? And that is reproduction. This differs from an endospore where one cell makes 1 endospore. This is a survival strategy. The parent cell will die, and so 1 cell makes 1 cell. That is not reproduction. It's a survival strategy. Any questions about any of that? So when you tell the word spore, you should clarify whether you mean an endospore or a reproductive spore. The textbook isn't always good about that. So most bacteria divide by binary fission. Let's go through that now. Where one cell will replicate its DNA molecule, and then one DNA molecule move to one side of the cell, the other DNA molecule will move to the other side of the cell. And then the cell will literally split down the middle, growing a cell membrane and a cell wall. Come on. Sorry, my mouse has died. There it is. Growing a cell membrane and a cell wall right down the middle of the cell, dividing the cell into two. Here is an actual electron microscopic image of a cell going through binary fission. And then the cells will separate and become separate daughter cells. Any questions about binary fission? Now you'll note that when the cell divides, it frequently will be smaller than the parent cell. So the newly replicated cell may enlarge before it will go through a division. That enlargement is not called cellular growth. It is called, well, enlargement. Because growth refers to, with microbes, an increase in the cell number, not an increase in cell size. All right, let's look at budding, where a cell right here can put out a little bud on one end, and that bud can grow and grow until it becomes about the size of the parent cell. And then the bud will separate from the parent cell and become its own independent cell. Let's watch a little movie on budding. Assuming this is going to play. Oh, there it goes. Just a little slow. This is interesting. Verify to continue. So we watch that cell there. It's going to put out a bud right there. And the DNA is actually actively dividing now. And right there, you can see the DNA moving into the bud. The bud enlarges. And when it becomes about the same size as the parent cell, the bud forms a cell wall separating itself from the parent cell. And then the parent cell can put out another bud right there. And the newly replicated cell or budded cell is going to start putting out a bud right there. Any question about any of that? All right. When we're talking about bacterial growth, we use the term generation time. Surprisingly, students get the generation time incorrect a large number of times. The generation time is the time required for a cell to divide, and it is the same time as for the population number of those cells to double. So when the population size doubles, that is the generation time. And then that's the time it takes for one cell to divide into two cells. This generation time can vary considerably between species. It can be as fast as 20 minutes, like E. coli can have a generation time of 20 minutes per generation. But most bacteria have a generation time between 1 and 3 hours. Mycobacterium leprae is the slowest bacterium I know, and it has a lot of mycolic acid. Making the mycolic acid is time consuming. It has mycolic acid, it puts on the cell wall. And all of the mycobacterium tend to grow slower because of the time it takes to make mycolic acid. But Mycobacterium leprae, the bacterium which causes leprosy, is the slowest bacterium that I know for its generation time, where it takes 10 days for one cell to divide into two cells, or for the population of Mycobacterium leprae to double in size. Now, what students get wrong on the generation time is, I think, because you're so used to talking about driving and miles per hour, we have a lot of things per hour. The generation time is not the number of generations per hour. It is the time per generation. And that per generation is frequently assumed and dropped off. Like when I say that E. coli has a generation time of 20 minutes, I mean E. coli has a generation time as fast as 20 minutes per generation. Any questions about any of that? So remember, the generation time is the time it takes for one cell to divide into two, or for the population number to double, which is the generation time. Any questions about any of that? All right. So when we're looking at the generation time and plotting it, microbiologists like to use a semi-log plot. And the reason for using a semi-log plot is because if you were to use a regular arithmetic plot, the graph would look something like that. One cell here will divide into two, two into four, four into eight, eight into 16, 16 into 32, meaning there's a whole bunch of points down here. It's hard to separate these points. And then the next point here won't be on the graph. It's going to be on the ceiling. So it's hard to put additional points on the graph, on the arithmetic scale. And then the other reason that you don't like it is this is the function for the arithmetic growth, and it's a complex mathematical formula. Something equals something like y equals mx plus n x squared plus b, and there might even be another one in there, another constant. When we use a semi-log plot, we get a straight line where the points here are better spread out. And then the next point there can actually go on this graph about right here. So it's off the graph, but it's still on the page. Okay. And then the main reason for using a semi-log plot is this is a very easy equation for explaining the growth, and that is y equals mx plus b. And there's a plot of the log, and there's a plot of the arithmetic scale. And note that this is not a log plot, it's a semi-log, meaning the y-axis is log scale. The x-axis is arithmetic. It's still the number of generations. Any question about any of that? All right. So that's why microbiologists like to use a semi-log plot. When we're talking about bacterial growth, we discuss them with a growth curve. Most organisms, as well as most microbes, follow this growth curve. In fact, they've noticed that deer on an island follow this growth curve too, meaning when you first introduced the deer to the island, the deer were exploring the island, they weren't reproducing, so they had a lag phase. And then when it became comfortable with the environment of the island, they then entered their growth phase. And then the deer population became stationary. The reason being is that the food supply started to become short, and so that the population numbers became stationary because for every new deer born, meaning a lucky parents that could have an offspring, there was a deer that was dying because of lack of food. And then as the environment of the island got worse, the deer population then entered the death or decline phase, where the number of deer on the island actually decreased. When we put bacteria from a solid culture into a liquid culture, we get this growth curve, where initially the bacteria don't grow, they're in a lag phase. Why are they in a lag phase? Because we've changed the environment and the bacteria have to make the enzymes and intermediates they need for growing in the new environment, meaning for growing in a liquid environment. Once they've made the enzymes and materials they need for growing, they then enter the log or exponential growth phase, where initially the microbes grow very rapidly because there's lots of nutrients around, not that many bacteria cells around, so there aren't many waste products around. That growth will begin to slow, and that's because there's more bacteria now, so there's less room for the bacteria to grow in. There's more waste products around, and there's less nutrients around. So the bacterial growth begins to slow. And it'll slow to a point where we enter the stationary phase. The lucky cells are the ones that have some nutrients around them, some space around them, so they can grow. And the unlucky cells have lots of waste products around them, very little, if any, space around them, and they are dying. And so in the stationary phase, the population number is stationary or remains about the same because the number of new cells equal the cells dying. So the population becomes stationary. Now, as time goes on, that environment will become less favorable. There will be even less nutrients, less space, and more waste products building up. And so the population will leave the stationary phase and they will enter the death or decline phase, where the population numbers actually decline. And that is because the number of new cells will be less than the cells that are dying. Now that will be early in the death or decline cell, where there will be some cells which are very lucky and they are still growing. But the majority of the cells will be dying, and that's why the population will decline. But late in the death or decline phase, there will be no cells growing, so there'll be no new cells, and all the cells will be either dying or will be stationary and not growing. The length of the death or decline phase until the population number becomes zero depends on the species. Some species like E. coli, you can go through this bacterial growth curve in about 2 weeks. And after about 2 weeks, 2 or 3 weeks, all of the E. coli will be dead. But other cells, especially cells that have endospores, they can live for years or centuries. Any question about any of that? All right, so explain the log phase, lag phase, where the cells are making the enzymes and nutrients they need to enter the growth phase. They will then be followed by rapid growth because there's lots of space, lots of nutrients, very little waste products around. And that will the growth will then decrease as the waste products build up the space becomes limiting. And what else? Oh, there's less nutrients around. And then as the environment decreases further, the population will enter the death or decline phase, where more cells are dying than cells are growing, if any cells are growing. All right, any questions about any of that? If not, let's talk about the different ways we have for measuring microbial growth. There are different ways to measure the growth in a microbial population. There are direct methods and there are indirect methods. Direct methods include a plate count, also called a viable plate count, the filtration method, the most probable number, and then a direct microscopic count. I've done all of these direct methods except for the most probable number. And then there are indirect methods for measuring the number of cells growing, and that's the turbidity, the metabolic activity of the cells, and then the dry weight of the cells. Let's talk about each of these. Plate counts are the most common method used in microbiology for measuring the number of cells. This actually measures the number of viable cells. So it is sometimes called the viable plate count. This takes 24 or more hours for the colonies to develop. Once you plate the cells, they have to incubate for 24 hours or longer. The results are often reported as the number of colony-forming units per mil of inoculum. We usually make a serial dilution when we're doing a plate count. So we have an original sample, and if the bacteria are too numerous in the original sample, which most of the times they are, we then dilute them in a serial dilution. And then we plate each dilution and then look at the colonies that develop on the plate. Many of the plates will have too many cells to count, and that can be abbreviated 2MTC or 2NTC. 2NTC stands for too numerous to count. And then there will be some with good colony numbers, and then some with few or no colony numbers. The ones you want to count are the plates with between 25 and 250 colonies on the plate. And if you have 25 to 250 colonies on the plate, you can then say that you have 25 to 250 colony forming units on that plate. We call it colony forming units because we do not know if the colony was formed from one cell, two cells, or a clump of cells. Plate counts are performed by using either the pour plate or the spread plate method. What you do is you make a serial dilution, meaning we have an original inoculum, and then we take 1 ml of that, put it into 9 ml, so we have a 1 to 10 dilution, mix that up, take 1 ml of this, put it into 9 mLs, we have another 1 to 10 dilution, for a total dilution of 1 to 100. Take a ml out of that, put it into 10 mLs, 1 to 1,000 dilution, take a ml out of that, actually mix it up, take a ml out. Then you have the 1 to 10,000, take a ml out of that, then you have the 1 to 100,000. Now you need to plate each of these dilutions either on a pour plate where you're putting in 1 ml of the dilution and then spreading that 1 ml that's been poured on the plate around on the dish to get the colonies to spread out. Or you can take 1/10 of a ml from the dilution and then spread that around with a spreader. You have to sterilize it and then cool it and then spread the 1/10 of a ml around the plate. And then that will spread the colonies around. Either way, you get colonies tending to be isolated in the right dilution. But many of the dilutions, like the original, the 1 to 10, the 1 to 100, will be too many to count or too numerous to count. This one will be too many to count as well. You might be able to get an estimate, but it won't be an accurate count because there's too many colonies fused together. And then you have a plate with the number of colonies between 25 and 250. You base your colony number on this one. And here we have a plate with the number of cells from this dilution being 4. We want to use this plate because the mean for the number of cells or colony forming units per mil of this dilution will be better defined than this cell. And let me explain that. Well, actually, let's go with Word. If you only have 4 colonies on the plate, that means the mean equals, well, let's go mean. Or plus or minus 1. I'm going to underline that. All right, so that's 4 plus or minus 25%, which is a huge number. I'm not going to do the plus or minus. Let's just put that in and copy it there. Whereas if we have our mean of 100 colonies per plate, the mean equals 100 plus or minus 1. Oops. Or 100 plus or minus 1%. And that's why we use the plate that has a count between 25 and 250 colonies per plate, because the mean will be much tighter. Plus or minus 1 is only plus or minus 1%. With 4 on the plate, plus or minus 1 is the mean is 4 plus or minus 25%. Any question about any of that? So we use a plate, this plate and not that plate, because our mean will mean something. It'll definitely be close to this number, if not this number. And this one will be this number plus or minus 25%. Huge variation compared to that one. Any question about any of that? All right, so this is the main way microbiology students obtain the number of cells in the original inoculum. And let's go ahead and calculate this. I blow that up so I can read it. So the dilution is 1 to 10,000 here. And the number of colonies, is that stated there, or are we just gonna guess?
I personally didn't see it stated there, but
Okay, let me take a look here. Then let's just
maybe.
get. All right, that looks like it's 30 to 40. Does anyone disagree? So we're just
I,
gonna say 40. So then the cells per mil in the original dilution will equal, whoops, I don't have that right, 40 colonies on the plate. 40 times the dilution, which will be equal to, what is that? 4, and that 10,000, 1, 2, 3, 4. So 400,000 if I did that right. All right. 400,000 cells, well, actually, colony forming units per mil. And this is assuming we're plating 1 mil, so we have to put that in there. So if there's 40 colonies per plate, the dilution's 1 in 10,000. The number of cells per mil in the original equals 40 times 10,000, or 400,000 colony forming units per mil. We say colony forming units instead of colonies per mil or cells per mil because we don't know if one cell formed a colony, two cells formed a colony, or a clump of cells formed a colony. Let me see if I can see a difference here. This looks like it's made from an artist, so it's not going to show it. Okay. All right. Any question about any of that? That is how you get a cell count when the original is very concentrated for the number of microbes, which many times in our real life world, this will happen. Occasionally in our real life world, though, the original sample will be very dilute. Like somebody who's testing drinking water and they want to discover how many microbes are in the drinking water. Or drinking water, at least in most cities, is pretty dilute. It does have microbes in it, but it's very dilute. And what you want to do is you want to filter the drinking water through a filter, catch the bacteria on the filter, and then put that filter on a plate. And then these cells will go on to form colonies on the plate. So whenever the number of bacteria is very low, you need to pass the bacteria through a filter and then grow the filter up on a plate.
Makes me wonder what's in my well water.
Uh,
Well, underground spring, it's a natural spring, but it makes me
yeah,
wonder now.
it depends. There could be some chemosynthetic bacteria in it, in which case there might be a fair number of bacteria in it. But if there is no chemosynthetic bacteria in it, then it will be fairly clean. Not perfectly clean, but fairly clean.
Better than any city water I've ever tasted.
Ah, yes, well water has that better taste. For one thing, they always put chlorine in city water to prevent microbial growth as well as decrease microbial growth.
I get it. When I go out to eat and they ask me if I want water, I'm like, no thanks.
Okay. All right, so that's the second method, a direct method for getting a microbial count. And both of these methods, you're forming colonies, so only the viable cells are being counted. Meaning if we were to plate a non-viable cell, well, let's go with this plate. A non-viable cell right here, it's not going to grow and form a cell. So we're only counting the viable cells. That differs from a direct microscopic count where you're counting all cells, both viable and non-viable. And you're using a microscope. And when you look at cells under the microscope, you cannot tell, at least most of the time, whether the cell is alive or whether the cell is dead. Any question about any of that? So a direct microscopic count uses a measured volume of liquid placed within a defined area on a gridded slide. That gridded slide has a specific volume under it, and then the cells are counted microscopically. It's not good for bacteria that are motile because the bacteria can move around while you're trying to count them. It does count both viable live cells and dead cells. A high concentration of cells are required, but you could centrifuge the sample to make the cells higher concentrated. And then no incubation time is required. You get your count directly when you finish counting under the microscope. This uses a gridded slide like this right here, and then you put the volume under this slide, cover slip, which only allows a certain volume there. And then you usually count one square here and one square there usually has 1 microliter of liquid underneath it, meaning the volume for this would be 1 microliter. And then you make your count here. What is that, about 15 to 20? And then you multiply by 1,000,000 because 1 microliter is 1 millionth. And they're showing the slide from the side. This is a fairly quick way of getting your numbers. Obviously, you're going to get both dead and live cells, but it's a little, what are you saying, a little hard on the eyes of the person doing the counting. Any question about that? All right. So those are the direct methods. I'm not going to talk about the most probable number. You can read about that in the textbook, but you're not going to be quizzed on it. I've never done the most probable number. It's not used very often in microbiology. There are indirect methods, and I've done all of these. You can do turbidity, metabolic activity, and dry weight. This is where you're estimating the number of cells. You're not getting a direct count of the number of cells. Turbidity uses a spectrophotometer where the liquid in a tube is first measured to see how much light comes through the liquid of the media. And then you measure the same tube and media with the cells in it, and the cells will block the light. So this light ray will get through, that light ray will not because the cell is going to block the light. You then measure the amount of light coming through. And then you look on a chart to see how many cells this measurement, how many cells per mil this measurement is. So somebody has to set up the chart by having different cell numbers, which usually they do by doing a cell count, having different cell numbers, and then measuring the turbidity, and then getting the number of cells for that turbidity, and then you make a chart. But once you have the chart set up, you can then use that chart for infinity to get the number of cells from the turbidity meter, meaning how much light is getting through. Any question about that? So usually you measure the absorbance, look on a chart. The chart will tell you how many cells per mil are in the tube. And this is a very, very quick and easy way to get your estimate of the cells. You just take the tube, put it in the spectrophotometer, measure the light, and then look up in the chart how many cells per mil are in the tube. Any question about any of that? Metabolic activity, I've used it a few times, not often, but it assumes that bacterial metabolic product will accumulate in direct proportion to the number of bacteria cells present. For example, if you're measuring the CO2 being produced, the more CO2 that's made means there's more cells. And so you can count the number of cells increasing over time by measuring the CO2 produced over time. Any question about any of that? You don't have to measure CO2, you can measure acidity or something else, but often CO2. Dry weight is a method which is convenient because you just grow up the cells, spin them down in a centrifuge, pour off the supernatant, then dry the cells, and then at your convenience, you weigh the cells. The more the weight, the more cells you have. Once again, you need to have a chart set up where somebody has actually counted the number of cells, weighed them, and set up the chart. But once you have the chart set up, you can use it for infinity, and you just spin down your cells, pour off the supernatant, dry the cells, then weigh the cells, and then look up on the chart how many cells you have. Any question about any of that? All right, that's it for chapter 6. Any questions about chapter 6? All right, let's move on to our next chapter. I think it's chapter 8. Close that down. Yep, chapter 8. Chapter 8 is genetics. You'll note that I've spent, the syllabus has chapter 8 for a longer period of time than most other chapters. It's because we are going to go into it in detail, and it's an important chapter. Chapter 8 is the genetics of microbes. My computer is slow for some reason today. Oh, there we go. The introductory slide is shown here. Always know the major goals and the rough lesson outline. So know the terms. What's a genome? What's a gene? What's a genotype? What's a phenotype? What is vertical gene transfer? And what is horizontal gene transfer? What is an operon? What is a promoter? What is an operator? What is a structural gene? And what is crossing over? Whoops, got a few more, 3 more. What's transformation, conjugation, and transduction? And then 2, be able to describe DNA replication, RNA transcription, and protein translation. And then lastly, 3, understand the different types of mutation. Any questions? All right, we're going to talk first about the structure and function of the genetic material. We'll talk about the flow of genetic information, talking about DNA replication, RNA transcription, and protein translation. We'll then turn our attention to bacterial gene expression. We will use the operon model to explain bacterial gene expression. We'll talk about what an operon is and what the various parts of it are. We'll then briefly talk about mutation, talking about the different types of mutations. Then move on to genetic transfer and recombination. And we'll end this lesson talking a little bit about genes and evolution. Any questions about anything? If not, let's get into the first terms. Genetics is the study of what genes are, how they carry information, and how that information is expressed, and how genes are replicated. A gene is simply a segment of DNA that encodes a functional product. That functional product is usually a protein, but it doesn't have to be. The functional end product encoded by the gene may simply be RNA. Any question about any of that? So obviously, if the gene codes for tRNA, rRNA, or some other form of RNA, mostly tRNA or rRNA, the gene will only encode RNA. But if the gene codes for a protein, messenger RNA will be transcribed from that gene, and then the messenger RNA will be read to translate the gene code into a protein. Any question about any of that? So here we're looking at a chromosome. This is a condensed chromosome. And there we're looking at it spread out, where you'll note that this region of the gene from sequence 1 here, come on, sequence 2, etc., all the way down the DNA. Nucleotides to here is gene 1. Then we have a region which is not a gene, and then gene 2. And this could be a eukaryote chromosome, which actually eukaryote in their DNA, most of the DNA does not code for a gene. That differs from a bacteria, where most of the DNA does code for a gene. And there's I think a human gene showing you the different location of the gene. I can't read this, but here's a gene here, there's a gene there, there's a gene here, which is almost on the same location. So that's muscular dystrophy. And this is chronic granulomatous disease. I can't read the other sign. Adrenal hypoplasia. Many of the genes are named after diseases because if the disease is caused by a gene, it will be a region that they call the gene after that disease. Any question about any of that? All right, let's move on. I thought I had another picture of a chromosome. Oh, here it is here. There's a chromosome coming out of an E. coli cell, And all it is DNA, one nucleotide after another, connected all together, making this very long string of DNA, which in bacteria, usually there's just one string or one chromosome in the cell. Any question about any of that? Now, the DNA is not alone. When we call something a chromosome, you should realize that it's DNA and protein. In eukaryotes, the major protein is histone protein, frequently just called histone. And the DNA molecule actually wraps around that histone protein. Actually goes twice around it. But in prokaryotes, or at least bacteria, there is no histone protein. There are other proteins in the DNA to make the chromosome, but there's no histone protein in bacteria. Any question about that? All right, some more terminology. Genome is all of the genetic material in a cell. When we're talking about a microbe, a genome could refer to all the genes in a organism because a microbe is a cell. The study of genomes is called genomics. Two terms involved with the genome is the genotype, and this is the actual genes of an organism. For example, the genotype of a blue-eyed individual is lowercase b, lowercase b. The genotype of a brown-eyed individual will either be capital B, or capital B, lowercase b. And brown-eyed allele dominates over the blue-eyed allele. And so an individual with one brown-eyed allele, meaning one brown-eyed gene, and one blue-eyed gene, lowercase b, will have a brown eyes. Any question about any of that? The phenotype is the expression of the genes. So the two lowercase b alleles, meaning lowercase b, that's the genotype, has a phenotype of blue eyes. So the phenotype is the expression of these genes. And for the individual that has capital B, the phenotype will be brown eyes or brownish eyes. But for the individual which is capital B, lowercase b, the capital B dominates over the lowercase b, and so the individual will have brown or brownish eyes. Any question about any of that? Now, when we're talking about phenotype, you should realize that we're talking about simple genetics. What else was I going to talk about with this? Just lost my mind, so I'm not sure. Anyways, this is simple genetics where, oh, I know what I was going to talk about, where the eye color is determined by one gene pair. Lowercase b is bluish or blue eyes, and then one capital B, and then either another capital B or a lowercase b will be brown or brownish eyes. Any question about that? And yes, our eye colors do not fall into simply blue and brown, but they do fall into bluish and brownish, meaning if you call very dark, almost black eyes to brown-green eyes, hazel, that is brownish eyes. And then all shades of blue, from blue-gray to blue-blue to green, either blue-green and then green, these are bluish eyes. Any question about any of that? All right. So the genotype, at least for our simple genetics, determines the phenotype. And there is genetics beyond simple genetics. We're not going to talk about it. But somebody's weight, for example, is not a simple genetic trait. And it's true, there are some genes that will determine your weight, but there's a lot of environmental influences which can determine your weight. For example, if you start eating a lot, you're going to weigh more than if you don't eat a lot. And so the environment can influence that trait, your weight. It's not a simple genetic trait. Any question about any of that? All right, I already talked about that, so let's move on. So DNA is a polymer of nucleotides. There are 4 nucleotides in DNA: adenine, or A, thymine, or T, cytosine, or C, and guanine, or G. And I don't care which of the names you memorize, you should know either adenine, thymine, cytosine, or guanine, or A, T, C, and G. DNA is a double helix associated with proteins, meaning that DNA, at least in our cells, is 2 strands, one running that way, one running that way. A double helix. And then our DNA has proteins in it that makes the chromosome. So a chromosome is always proteins and DNA. And in eukaryotes, the main protein will be a histone protein. There are other proteins, but the main one is histone protein. In DNA, we have 2 strands, a single strand of DNA running this way, and another single strand of DNA running this way. The single strand of DNA is held together by the deoxyribose phosphate backbone. That's the sugar phosphate backbone. And this is a chemically covalent bond holding the single strand of DNA together. Any questions about any of that? Now, the 2 strands of DNA are only held together by hydrogen bonds. The hydrogen bonds occur between the nucleotide bases of one strand and the corollary nucleotide base on the other strand. So G will hydrogen bond with C, forming actually 3 hydrogen bonds. And that's true for C hydrogen bonding with G. And then T will hydrogen bond with A, and then A with T. And there will be 2 hydrogen bonds in the AT connection. The 2 strands of DNA are anti-parallel. That means one runs this way and the other one's that way. I don't know if you can see that. I'm trying to show you. One's running this way and the other's running that way. You don't really need to know, but this is the 3' end. Let me get that out of the way. This is the 5' end. This is the way chemists number the carbons in the sugar here. This is the 3rd carbon in the sugar, and that's the 5th carbon in the sugar. And that's why this is the 3' end, and then this is the 5' end. All right, when we're talking about the flow of information within a cell, there is the vertical flow of information within a cell, and this is known as the central dogma, where the information in DNA flows first to RNA in transcription, and then the information in RNA flows to protein by translation. And this is known as the vertical flow of information within a cell. You should know that DNA, RNA, and proteins each have their own language where the information is stored. DNA, the information is stored in the deoxyribonucleic acids, meaning the deoxyribonucleic nucleotides, which is then stored in the RNA, which doesn't use deoxyribonucleic acid, it uses ribonucleic acids. So it's a similar language, just with a different dialect. And then the information is translated into the amino acids, a protein, and that would be a totally different language. So that's the flow of information within a cell. Remember, we do have the flow of information between generations of cells, where the information in the parent cell flows to the information in the daughter cells. How that happens is the parent cell replicates its DNA, making a clone copy of its DNA, and then one DNA molecule moves to one side of the cell, the other DNA moves to the other side of the cell, then the cell splits down the middle. So each daughter cell gets a copy of the information in the parent original cell. You could talk about that in humans, in which case the information flows from two parents to one offspring. And it's just that humans have sexual reproduction. For why it's different. Most bacteria reproduce asexually, why one cell can divide into two cells. Any question about any of that? We also have the flow of information between cells of the same generation. Or in other words, where DNA leaves the original cell, the donor cell, and then that DNA moves into another cell that we call the recombinant cell. And this cell can express genes that were in this cell, the parent cell. Usually, but not always, the parent cell will die, releasing the DNA into the environment, and then the DNA will be taken into the recombinant cell. And that's why I've broken this graph to make it a little clearer that I'm talking about the DNA leaves this cell, is then free living, or free DNA. I don't know if it's living because it's not outside of a cell. And then it can be taken into a recombinant cell. And then the gene coming from this cell could be expressed in this cell. Any question about any of that? All right, I've already talked a little bit about this. Let me talk about the central dogma in a little more detail. The DNA, the information is stored first in DNA, it's copied and transcribed into RNA, and then the information in RNA is translated into the information in proteins. The language of the DNA are 4 DNA nucleotides. So the number of letters in the alphabet is 4. And then the information is stored in the ribonucleic acids, once again in nucleotides, but they're different nucleotides than that nucleotide. Once again, we only have 4 letters in the alphabet because RNA only has 4 nucleotides. Three of them are the same as DNA, and that would be C, A, and G. So guanine, adenine, and cytosine. Those nucleotides are the same in DNA and RNA other than the sugar, which in DNA is deoxyribose and RNA it's ribose. And then the information in the RNA is translated into protein. The language are the amino acids. There are 20 different amino acids, at least in humans. So there's 20 letters in this alphabet. And for comparison, English, and a language all of you are familiar with, has 26 letters in it. Any question about any of that? So if the information in DNA was coded for "that is a dog," the information transcribed into RNA will be something similar, just a different dialect. That is a dog. And then the information will be translated into protein, and it would be translated something like quello è un cane. The original information, but in a different language. Any question about any of that? All right, let's talk about the flow of genetic information between generations of cells or between a parent and its offspring. This is called the vertical flow of information, where the information flows from parent cells to daughter cells. Vertical flow of information. When we're talking about the flow of information between parent cells and offspring cells, DNA replication is how the DNA moves from parent to offspring. First, the strand of DNA has to be copied. DNA replication is semi-conservative. What that means is that one strand is conserved from the original strand, and then a new strand is made to make the new double helix of DNA. So DNA replication is semi-conservative. One strand is used as a template to make the new strand, and the polymerase sitting right here just looks and sees that there's a G there. Let me blow this up to make sure I got that right. Yep, a G there. So it knows to add a C there because C is complementary to G. Only a C can be added here, at least under normal circumstances. Any question about any of that? Then the RNA polymerase moves to the next nucleotide. It sees a C there, so it will only add the nucleotide G for guanine, because G and C pair with each other. And then it'll move down the messenger RNA. Not messenger RNA, sorry, this is DNA. Move down the DNA and see that there's a there, and then put on the correct nucleotide here, which would be a T. Any question about any of that? So semi-conservative means we have our original strand of DNA, which is black. And when this strand is replicated, it'll be split into two separate strands. Each strand will be used as a template to make the other strand. And then the newly replicated DNA, which is double-stranded, will contain one strand of the original RNA and then one newly replicated strand. This is fairly important for life because if the RNA polymerase put on the wrong nucleotide, the cell could find that error and then remove the incorrect nucleotide. And then another enzyme will bind to this DNA and add the correct nucleotide. It is not photosystem 1 of photosynthesis or chemosynthesis 1. It's some other protein, which is well, you don't need to know that, so we'll skip it. And then the other daughter strand will be the same, where one of its strands will be the original strand of DNA, and the other will be the newly made strand of DNA. This is called semi-conservative replication. Conservative replication, which doesn't happen, would be when this DNA replicates, one strand would have two dark or the original strands of DNA, one cell, and then the second cell will have very little, if any, of the vitamins. And the reason why it's very little, if any, is that vitamin C is not very stable. If you simply heat it up, bring it to a boiling temperature, you destroy the vitamin C. With elevated temperature, you begin to destroy the vitamin C, but not all of it will be destroyed. Vitamin C also has a, what do you call that, a time constraint. So if you buy a bottle of vitamin C, you should try to use it shortly after the expiration date, because vitamin C, even in the airtight packaging, will decrease with time. Come on, I'm trying to get to paint. Oh, there it is. So with time, vitamin C will decrease. And this is not very long. Obviously, it'll stay around easily for more than a day. So when you eat your fruit or something to get vitamin C, it will be in your body for at least 24 hours. But once the threshold drops below a certain number, oh, don't have that right. You then need to take more vitamin C to bring up the level of vitamin C that's absorbed and digested to a high enough level. So usually you want to be taking vitamin C in this area. You don't want to be taking it below this area. Looks like I can't draw with that. Come on, mouse. I want the vitamin C in this area. Not on the extremes. All right, any question about any of that? So once again, looking at the strands of DNA, the two strands will separate. And then this strand will be used as a template to make a newly made strand of DNA. So the nucleotide will C and A here, and then add a T for this strand of DNA. And here we have a C, the RNA polymerase will add a, excuse me, this is DNA polymerase will add a G here, because G binds to C. Any question about that? One thing that you don't really need to know, but when we're adding the nucleotide, we're actually adding a triphosphate of that nucleotide. So for thymine being the DNA base, it's going to have a deoxyribose and then 3 phosphates. 2 of the phosphates come off, meaning this bond here is broken, releasing energy. That energy is used to bind this phosphate to that deoxyribose. So that energy which is released is used to attach this newly added nucleotide to the growing new strand of DNA that's being replicated. Any question about any of that? Remember that 2 strands of DNA have 2 single strands of DNA held together, and the single strands of DNA are held together by the sugar phosphate backbone. Sugar, phosphate, sugar, phosphate, sugar, phosphate. This is a very high bond, very strong. Can be broken like in aerobic respiration, but oftentimes humans have trouble breaking this bond. All right, I did mention that this template can hydrogen bond to this template. So this template is, and this going in this direction, this template is going in that direction. I'm not talking about how it's growing. I'm talking about the 3' and the 5' end. All right, any questions about any of this? If not, let's watch a little movie on DNA replication.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about that video? It's actually quite good and went through a lot in a little bit of time.
I wish they'd use that video in previous biology and chemistry classes. I like that. Thank you.
Okay. All right, let's talk about DNA replication in a little more detail. We'll not really go into more detail than the video went into, but DNA is copied by DNA polymerase. The new DNA is always made in the 5' to 3' direction. It's initiated by an RNA primer. I'm not going to quiz you on the RNA primer, but there are two strands of DNA. One is called the leading strand, and it's running in the correct way. So the DNA polymerase just moves down the DNA, synthesizing new DNA in the 5' to 3' direction. The other strand of DNA, called the lagging strand, this DNA is running in the incorrect direction, meaning the DNA polymerase cannot bind here and then move down this way because DNA polymerase has to move that way on the DNA. Is that clear to everybody? And that strand is called the lagging strand. Let me see if I can blow this up a little. So how the lagging strand replicates itself is that the DNA polymerase makes short batches of DNA kind of running in the wrong direction. It's running in the correct direction, but in the opposite direction that the helicase is moving. Let me state that there are 3 enzymes needed, absolutely required for DNA replication. That is DNA helicase. It binds to the DNA and then opens up the DNA, converting a double-strand DNA to single-strand DNA. Only the single-strand DNA can be replicated, and that occurs by DNA polymerase. The second enzyme that's required for DNA replication is DNA polymerase. It binds to the original strand of DNA, or the old strand of DNA, and then reads it, and then adds the correct base, complementary base, which will be added to the growing new strand of DNA. And then the third enzyme is ligase, and it hooks the DNA together. I'll talk about ligase in a little bit after I've finished talking about the leading strand and the lagging strand. Now, for the leading strand, if the DNA polymerase is lucky, it could begin on this end of the chromosome, and it may copy the DNA all the way to the end of the chromosome. That would be if the DNA polymerase is lucky. In reality, the DNA polymerase happens to fall off every once in a while because it's binding to the DNA just by a stoichiomic reaction. And every once in a while, the DNA polymerase falls off. Let's say it goes to here and then it falls off. That doesn't mean the rest of this DNA isn't copied. It's just that another DNA polymerase has to bind right here and then continue replicating that strand, the leading strand of DNA. In which case there will be a gap right here between the, I guess, the first new strand of DNA that was made by the first polymerase, and then there will be a gap between the newly replicated DNA made by the second DNA polymerase. It's ligase that comes in and makes a chemically covalent bond between the DNA of that strand, which is a new strand, and this second new strand of DNA. You understand what ligase does? So ligase doesn't do very much on the leading strand. Only when the DNA polymerase falls off, and then obviously a new DNA polymerase begins synthesizing this strand again, the ligase will have to put one time a link for each time there's a fall off of the DNA polymerase. And if the DNA polymerase doesn't ever fall off, ligase doesn't ever work on the leading strand. On the lagging strand, though, ligase is going to work an awful lot, because on the lagging strand, the DNA will be made in short batches called Okazaki fragments. This is named after Dr. Okazaki, and I could be mispronouncing his name, who discovered it. So the DNA polymerase moves this way, and then this DNA polymerase moves this way, and each Okazaki fragment will need ligase to connect it to the nearby Okazaki fragment 2 times on each end of the Okazaki fragment. And there will be a lot of Okazaki fragments on the lagging strand of DNA, because the DNA is moving this way and the helicase is moving that way. Any questions about any of that? Hopefully I said the RNA polymerase is moving, I mean the DNA polymerase is moving that way and the helicase is moving that way. And here you can see there's the Okazaki fragment, and there is the newly replicated strand of DNA, which this would be the nearby Okazaki fragment. And the DNA ligase binds there to connect this strand of DNA to that strand of DNA. Any question about the 3 enzymes that must work, you must know, for replicating DNA? The helicase, the DNA polymerase, and then the ligase, DNA ligase. There are some other enzymes that work. We're just not going to talk about them. I'm not going to go into the making of the primer and the removal of the primer and then the filling in the nucleotides where the primer used to be. Those enzymes have to be there too, but we're not going to talk about them. Any questions about the leading or the lagging strand and how DNA is replicated? I think that's it. Let me see if there's anything else I need to make. So new DNA is made only in the 5 to 3' direction. It is initiated by an RNA primer. I'm not sure why, but DNA cannot start the process of DNA replication. It has to be started by an RNA primer. And then that RNA primer has to be removed and filled in with DNA nucleotides. The leading strand is synthesized continuously. If the DNA polymerase never falls off the leading strand, the leading strand will be made in one strand. If it does fall off, it'll be made in 2 or more strands of DNA. If the lagging strand is made in many different chunks of DNA called Okazaki fragments. So we say the lagging strand is made discontinuously. And then I don't have on here. And then the ligase ligates, meaning hooks the 2 strands of DNA together, wherever there are 2 strands. such as in the two Okazaki fragments. All right, any questions about DNA replication? If not, I think I'm going to stop here. We're going to stop a little early, and I will continue at 7 for the lab. Any questions at all about chapter 8? All right, I'll see you at 7, oops, for the lab. Bye.